Hello everyone, and thank you so much for joining us today for our virtual launch. I hope you are all as excited as I am for what's to come in the next 30 minutes. I'm Vivian, I am responsible for scientific communication at Dynamic Biosensors, and I will guide you through today's event. Molecular interactions. They are the very essence of biological processes. They control everything that happens inside our bodies, whether in health or in disease. And since they are so crucial and complex, we at Dynamic Biosensors made it our mission to make it easy for you to study them. So for those of you who don't know us yet, we started out in 2013 and developed a technology called SwitchSense to measure interactions of biomolecules. In 2020, we already launched the second generation of our biosensors, the Helix Plus, and we can now draw back on 10 years of experience in real-time kinetic analyses and biosensing. The one question that came up again and again from our customers at conferences or in other discussions with scientists was, can you measure binding in real time on living cells? So we improved our technology, more than one ligand on the sensor surface, ligand density control, nanotechnology for ternary interactions. Our sensor surface was getting closer and closer to mimicking the real cell. But it still wasn't the real thing. Can you measure binding in real time on living cells? The answer was still no. Others also tried to fulfill this need. ELISA, flow cytometry, SPR-based biosensors. These technologies exist for quite some time now, and yet they didn't seem to do the trick, since they are not widely used to measure real-time kinetics on cells. So we started searching for a new solution. And after years of development, we found it. And it is surprisingly simple. So, can you measure binding in real time on living cells? I'm very happy to answer this question today. Yes, we can. And it only takes about 30 minutes hands-on time and 3 simple steps, thanks to the newest addition to our product portfolio, the heliXcyto. The heliXcyto is the first ever biosensor able to perform real-time interaction cytometry. So now I would like to introduce you to 2 very important people who played a decisive role in the development of this innovative product. Dr. Nena Macheco is the team lead of the R&D team for cell applications, and she developed this technology in the last years together with her team of outstanding scientists.
Thank you, Vivian. I'm really looking forward to presenting you this technology today.
Dr. Sebastian Letschert is the team lead for product development, and he coordinated the technical realization in order to design a biosensor instrument and the software.
Thanks, Vivian, and hello. I'm happy to be here today.
Now, without much further ado, I'm going to hand over to Nena, who will now tell you why and how to measure binding in real time on living cells. If you have any questions during the presentations, please feel free to use the chat function, and we will answer them later on in our Q&A session.
Okay, let's get started by taking a look at what you can do with RTIC. So RTIC is giving you access to interaction studies of various molecule classes on cells. And this includes small molecules binding to GPCRs, it includes antibodies binding to their cognate receptors, it can be a T-cell receptor interacting with a pMHC, or it could be a chimeric antigen receptor binding to its cognate target. All these interactions are characterized by a specific on rate and off rate, and these are resulting in an affinity and potentially an avidity KD. Real-time interaction cytometry is now the study of these association and dissociation rates of molecules binding to cells in real time. From the exponential increase and decrease of fluorescence during the association and dissociation phases, we can derive their respective on rate and off rate. The ratio of these values is the often used equilibrium dissociation constant, or commonly also referred to as the affinity of an interaction. So this technology will deliver biologically relevant interaction data of molecules binding to targets on cells. Let me point out to you next why interaction rates should be measured directly on cells. Imagine your target is, for example, a membrane receptor. To be functional, this receptor has a specific protein folding, which is strongly affected by the lipid molecules next to it. Furthermore, there is not one single receptor protein in the membrane, but many. The exact expression level depends on the target cell. But not only the overall density in the cell is relevant, but also the mobility of the receptor in the membrane. And last but not least, a native cellular membrane is full of different proteins, lipids, modifications such as sugars, and all these co-interactions by these molecules can have an enhancing or blocking effect on your actual interaction. If you take all this away, you lose a lot of important in vivo information. So some might wonder why they would need another technology if they can already check the affinity or the potency of a molecule on cells. These are probably important downstream assays, but you do lose a lot of information by jumping over the individual steps that are leading up to your potency and affinity. The association and especially the dissociation rate have proven to be a really predictive value that can be derived from in vitro assays before moving on to the expensive assays in animals or humans. By now, many scientific studies have proven that it is not always the strongest binder that is the most effective. For example, a long residence time of an antibody-drug conjugate might give a high on-target binding and uptake, which is good. However, this comes at the cost of a high off-target binding as well, and therefore side effects. So maybe you want to actually aim for something in the middle. Looking at the example of a cellular therapy, for example, with a CAR T cell, we know that a strong binding, of course, is increasing the efficiency in recruiting and activating a T cell. But at the same time, if a T cell is overstimulated, you actually result in an even lower immune response. So overall, again, you want to aim for something in the middle. Only if you really know the off rate, you can now target your drug to the dissociation rate that you really would like to have. And with that, I would like to hand over to Sebastian to give you more the technical details of how this technology can solve these questions.
Okay, thanks, Nena. So I'm going to start with a short overview about the current technologies on the market that are available for characterizing biological interactions. And what we do see here is that we have a degree of biophysical information and biological relevance. And if we have a look at technologies like BLI, SBR, and SwitchSense, so molecule-to-molecule interactions, they generate a huge extent of real-time information. So we can measure kinetics, but on a molecule-to-molecule level. On the other side, if we want to characterize interactions on cells, there are current methods like cell ELISA, plate assays, and flow cytometry, but they lack the real-time information. And so they are based on a single time point. So real-time interaction cytometry combines both, though a very high degree of biophysical information and biological relevance. Okay, let's come to the instrument, the heliXcyto, the new product which is made for measuring binding kinetics on cells. And the heliXcyto comes with a huge range of features, but also with consumables like chips and labeling kits and a powerful software package. And now I would like to go through all the features and starting with the optics. So the heliXcyto is equipped with a built-in reflected light microscope and a CCD camera for cell imaging. It has a two-color fluorescence detection for monitoring two processes at the same time, and it has super sensitive fluorescence detection with time-correlated single photon counters, which enable the analysis of low membrane protein expression levels. And there's a second spot for real-time referencing. So in total, there are 4 fluorescence detection channels and 1 CCD camera for cell imaging. If we have a look at the fluidic system of the heliXcyto, we see 3 fluidic lines which are connected to the chip from both directions. So we have here the sample and buffer line, so where the cells, analytes, and buffer is injected onto the chip, but also the wash line which comes from this side, which is optimized for parallel washing during dissociation, for example. On the other side of the chip, we have a regeneration line, a reverse flow over the chip, which pushes out the cell out of the trap, and by that regenerates the chip for the next run. The heliXcyto comes also with a high performance microfluidics for fast and slow kinetics, and the flow rates can be scaled up to 500 microliter per minute for artifact-free measurements of rapid kinetics. The material is very robust, that the heliXcyto is compatible with complex media. The autosampler is fully automated, and there is a fully automated liquid handling system in the instrument. And the sample tray, which is depicted here, can take several glass vials, but also 3, 84, and 96 well plates. The samples inside the autosampler can be temperature controlled between 4 and 40 degrees. And before the cell suspension is taken up into the liquid handling system, there's an automated cell resuspension that basically verifies that the cell density stays constant all the time. The heliXcyto automatic chip loader has space for 5 chips, which can be automatically exchanged in between assays. You also can decide whether you want to run measurements on a single or on multiple cells, dependent on the chip type. All chips are equipped with an RFID tag for seamless traceability, so information is stored after the measurement onto the chip, for example, the number of regenerations. The sample temperature during a measurement can be adjusted between 15 and 40 degrees, which makes it very easy to measure on living cells. Let's come to the heliOS user software. Several helix units can be connected to the heliOS network and can control by just one computer. So it doesn't matter if there is a Helix Plus or a heliXcyto device in this heliOS network. So with one heliOS instance, you can control as many helix units as you would like. The instrument has an embedded control PC for autonomous operation. So once a measurement is started, the instrument operates completely autonomous. heliOS, the powerful software package to plan, perform, and analyze RTIC, but also switch sense experiments, is shown here. And you have a set of verified RTIC methods which are easy to use and which supports an easy assay development. There's also an option to see the cell image snapshots taken during the measurement for visual inspection and for a quality check of the cells. We offer a set of automated data analysis where you can get to your fits and the kinetic rates, affinity, and avidity information in just a couple of clicks. Like you see here in this user interface screenshot, where we see 3 concentration of an analyte binding to a target inside a cell membrane and 1 long dissociation. It is also ready for batch analysis. What you can see here in this gallery view, where we have several interactions, and you can click through and get all the detailed information and the rates, which are also ready for export directly into a report or for further processing. So now I would like to hand over to Nena, and she is going to show you how to generate these data.
Okay, thank you, Sebastian. Then let's take a look first at what do we need to do in the lab actually to get an RTIC measurement done. And this is really simple. So you start by preparing a dilution of your fluorescently labeled analyte to whatever concentration is necessary, and you have the support of the heliOS software in deciding that. Next, you will harvest your cells from the cell culture. So that means you're just taking the cells out from a suspension solution, or you detach your adherent cells, and then you wash the cells and count them. Optionally, you can fix them if you want to run longer experiments on these cells. And then you place all your samples into the autosampler compartment of the heliXcyto, and basically you can start your measurement. The measurement itself is taking place on the heliXcyto chip. As Sebastian mentioned before, you can put several chips into the heliXcyto, and each chip has one single flow channel. Each flow channel has 2 electrodes, and one of these 2 electrodes is functionalized with small cell traps that you can see here. The cell traps are made from a biocompatible polymer so that we ensure that cells are not harmed during the measurement. The traps are of course flow permeable and they're stable in high flow rates up to 500 microliter per minute to ensure a technically accurate measurement. And because the traps are really just a physical barrier in our measurement channel, we are completely agnostic to what type of cell you want to measure with. So you can use living cells or fixed cells, you can use suspension or adherent cell types, and you can use primary cells or overexpression cell lines. It's really up to you. The first step of the automated measurement in the heliXcyto is the loading of the cells from the autosampler to the cell trap. So that means the autosampler is resuspending the cell solution, picking up 35 microliter and injecting it to the flow channel of the chip. And the cell will simply hit the trap and be immobilized in there. This capture event is controlled visually by taking automatic snapshots of the traps, and you can also judge the quality of your cell, if it is still living and healthy or maybe already an old fragmented cell. Then we come to the actual measurement. So we inject the fluorescently labeled analyte and allow the analyte molecules to bind to the cell. And this we can do at either one single concentration or if you want at consecutively increasing concentrations of analyte. Next, we come to the dissociation phase where we simply buffer continuously over the cell, and whenever a molecule is dissociating from its target, we wash it away, and by that we have a decrease of fluorescence that is giving us the dissociation rate of this interaction. The measurement itself has finished with this part, but we finish off the workflow by unloading of the cell to the waste. That means we are reversing the flow in the other direction, and by that wash out the cell and take it out of the trap, and this regenerated trap can be then used immediately for the next round of measurements, and we just restart the cycle. And again, of course, there are visual control snapshots that you know that your cells were in the trap until the end and have been removed from the trap after the regeneration. So after this introduction to how RTSC actually works, we want to, of course, see some real data. And this is what I'm going to show you now with the example of several antibody binding to different classes of molecules on different cell types. We'll get started with the category of immuno-oncology because this covers really a range of different molecules from antibodies to different cellular therapies. And I would say the most important category of the immuno-oncology are the antibodies from the checkpoint inhibitor class. And this is because when we look at the list of targets of antibodies in development, we see the PD-1, PD-L1 interaction complex at the very top. So most antibodies are developed for this interacting complex. What is PD-1, PD-L1? So you find normally in a person, you find PD-1 on T cells of our immune system, and PD-L1 is often expressed by cancer cells, because this interaction of PD-1 with PD-L1 is actually blocking the T cell from activation. That means the T cell, even if it would recognize the cancer cell, it cannot kill it. And now, of course, clever scientists have found a way to prevent this by developing antibodies that can bind either the PD-L1 or the PD-1. And whenever there's an antibody in the way, this interaction cannot take place and the T cell can still be activated and kill the cancer cell. One antibody that has been developed is nivolumab. This is an anti-PD-1 binding antibody, and we are now going to look at the on and off rate of nivolumab to PD-1. First of all, of course, we need to immobilize a cell that has PD-1 in its membrane. We used Hu7 cells, which are a hepatocell line, and as you can see, they are pretty large, so one single cell on our surface is giving a pretty good signal already. In the measurement trace, you can see here the real-time kinetics of nivolumab. So we measured two increasing concentrations in the association phase, and then along a dissociation that can be fit to reveal the affinity of nivolumab to be in the low nanomolar range, actually. And this fits very well with the therapeutic capacity of this antibody, that we know it works when we put it into a patient. It is binding stably to PD-1, is inhibiting this interaction and allows the immune system of the patient to kill the cancer cell. Another category that is very important are the complex membrane targets. And this is including, for example, GPCRs, ion channels, or multi-domain complexes. One example of multi-domain complexes is the T cell receptor. And the T cell receptor is really important for the activation and signaling during a T cell activation. For this reason, it is actually the top single target. So when I'm not talking about a complex, a PD-1, PD-L1, but as a single protein, then the CD3 protein, which is part of the T cell receptor complex, as you can see here, is the most targeted antibodies in development. The first antibody that was launched to the clinic already in 1986 actually was an antibody targeting CD3. It is the muronumab or OCT3 antibody. And again, although this has been in use in the clinic for a really long time already, the real-time association-dissociation kinetics of this antibody directly on cells have never been measured before. So of course, this is a challenge for us, and we wanted to do that. We measured 3 increasing concentrations of OCT3 antibody binding to, in this case, Jurkat cells, which are a category of T cells that, of course, have a T cell receptor complex in there. And we see a nice binding behavior increase of signal, and then followed by a very slow dissociation. So when we fit this slow dissociation, we reveal a nanomolar to picomolar binding affinity of this antibody. Again, a range where we know that this will work well in the clinic actually. Let's take a bit of a closer look at this dissociation here. So when we are measuring a dissociation, we are trying to find a fit model that optimally describes the data that we can see. And in this case, what we need to fit this data optimally is a biphasic fit. That means it is a fit that consists of 2 mono-exponential parts, and we can then assign one of the 2 parts, the faster one, to the affinity of the antibody, and the slower one to the avidity, as I will explain in the next slide then. This interaction was again measured one single T cell, in this case, again, a Jurkat T cell. So when we measure this multiple times on Jurkat cells, then we actually find that reproducibly we're getting 2 rates. So we always have a faster rate and a slower rate. The faster rate, from our knowledge already from switch sense experiments, normally is applicable to an antibody being bound with only one arm to a target. So it is a one-to-one binding mode. It is giving an affinity of this interaction. At the same time now, we also have measured a slower rate, and this is then applicable to an antibody being bound with 2 arms to its targets. So this is the avidity binding mode, a 1-to-2 binding. Interestingly, this is what we observed always on fixed cells and on some living cells as well, but we did see a bit of a difference between living and fixed cells, as living cells seem to only have very little of this fast dissociation rate. And we believe that this could maybe be explained by the mobility of the target in the living membrane, which means that the antibody snaps on with one arm to one target, but then a second target has the time to come into close proximity, and the antibody can bind with its second arm and eventually be bound with 2 arms in a very strong binding, resulting in this slower dissociation rate that we see here. Another important category of antibodies that we would like to mention are bispecific antibody formats. So bispecific antibody formats are used extensively in development, and at all times now we are increasing numbers of different formats, from large IgG types, multivalent binders, multispecific binders, fragments like FOBs or bytes or whatever. So this is really increasing, and there is a high need for advanced technologies that can actually assess the difference in efficacy and binding modes. Bispecific antibodies can be classified into 2 categories. The one category would be bispecific antibodies that have 2 binding sites against 2 different antigens on the same surface. That would be, for example, the therapeutic antibody Hemlibra that is binding 2 proteins of the blood coagulation cascade immobilized on platelets. The other category would be a bispecific antibody that can bind to antigens, but on different cells. And this, an example for this category would be a BiTE, a bispecific T cell engager that binds with one arm a target on a T cell and with one other arm a target on a tumor marker, on a tumor cell. And this is what we have measured now in this example. So this is BiTE molecules binding to a tumor marker in different tumor cells. And the example that I have here are 3 different bytes. And when we look at the rates of them, we can see that byte A and B have pretty comparable on rates and a bit of a difference in the off rate, whereas the byte C is actually not showing any interaction, and that fits well because the byte C is the negative control and we don't want to get any interaction from this one. For the byte A and B, when we look at the on rates, we see that they are pretty similar actually. And the difference in affinity that we see is coming mainly from the dissociation rate. So the byte B is binding stronger by a factor of 4 and therefore has the longer residence time on the tumor cell. The next example that I would like to show you is G protein coupled receptors, GPCRs. So GPCRs are a really important category of proteins because they are already targeted by approximately 30% of our approved drugs now. And that is surprising because it could be even more, as there are still like 60% of our GPCR not even targeted yet. So there are a lot of proteins out there that could still be used for therapies. But it is very difficult to work with them. The problem is that they most of the time have a very low native expression level, and it is really difficult to express them recombinantly and purify them and then still have them in a native setting. So what is necessary with these proteins is to really keep them in their target cell, in the membrane, and not extract them. This is perfect for us because we simply take the cell line as it is expressing a GPCR, and for this example, we received 3 cell lines with different expression levels of the same GPCR, so a low, medium, and high expression level. And then we were binding an antibody to these cell lines in the same conditions, and we were comparing the levels of fluorescence that we receive. See here, so the 3 different cell lines immobilized on the surface, and then we count the absolute fluorescence that we get from the antibody being bound on these cells and break it down per cell. And that gives us this plot here where we can then clearly see that the low expression cell line is expressing the target 12 times lower than the high expressing cell line. And this is very interesting for us because the feedback that we received in addition to this project was that the low expressing cell line could actually not even be detected anymore with a classical kit that is available for fax devices. So our sensitivity of the heliXcyto is good enough to also detect low expressing targets. Lastly, I want to mention some more projects that we have been working on, but there's not the time to show everything in detail. But I do want to mention that RTSC is also perfect to work on CAR T cells, so the interaction of chimeric antigen receptors with their proteins, or CAR NK cells also. We also have been working with T cell receptors modified or selected for certain PMHC molecules. You could look at the viral receptor binding, and of course we are also looking at small molecule binding, for example to GPCRs or other targets, including drugs or hormones, for example. And with these ideas, I would like to stop my presentation now and hand over to Vivian. Thank you.
Yes, the heliXcyto, biosensor meets cytometry. We hope that this overview could spark your imagination of what you can do with real-time interaction cytometry. And you can now send your questions to the Q&A chat, or you can also use the raise hand function and just ask Sebastian and Nina directly. So I'm just going to start and read out some questions from the chat we already received. So there's a first question that says, what is the minimum size for cells to be captured? Does it also work for bacterial cells, which are 0.5 to 2 micrometers in size?
I guess I will answer this question. So the size of the cells that we can capture depends basically on the size of the traps that we use, right? And we have different sizes of traps. So we can capture a pretty broad range of different cells. And I would say roughly we can capture from 6 micrometer on to about 25 micrometer. Everything that is smaller than a 6 micrometer, I have to say we would need to try, because it also strongly depends on the flexibility of the membrane of the cell. If it in the end can squeeze through the little windows, like the meshwork of the traps, and it could work for smaller cells, but to be honest, with a bacterial cell that is below 2 micrometer, I would say that this is probably too small and it's probably just going through the meshwork. So I would maybe not use RTIC at the moment, as we have the traps right now for bacterial cells.
Okay, I hope this answers your question, and I will just go on to the next one. The next question is, what is the limit of detection for Ka and Kd?
Okay, that will also answer that question, or if Sebastian wants to say something about that. So basically the limit of the on and the off rate that we can measure or resolve depends largely on the flow rates, like how fast can we exchange the fluidics on the surface, right? And we can pump with pretty high flow rates. We could pump up to 500 microliter per minute. Typically, we are using on cells somewhere between 25 and 100 microliter per minute. So that is still, as you can imagine, a pretty fast exchange of the analyte and the buffer, or the buffer and the analyte, depending on which phase we are. And we can resolve basically associations and dissociations happening within a few seconds. So this is like really fast process taking place. And we can still see that because, I mean, we're just continuously pumping over the surface. And I would say typically we're resolving fast dissociation rates in the range of 10 to the power of -2. I think we could go still a bit lower, but this is like the typical rates that we measure quite a lot now on the faster side. And for the on rate, I would say, I think we have not even reached or like practically reached our limit, because most of the samples that we receive that bind to targets directly on cells are rather slow binders, more like in the range of 10 to the power of 4 or maybe even 5 or something. But I think we could be much faster there technically, actually. So it really depends on the sample what we get.
Yeah. So now we get a little bit into the data analysis and how the data looks like. There's a question about the gap in our data, like why is there no data between 5 minutes and 10 minutes?
I take this one. So we have between the injections of analyte, there are liquid handling steps, for example, the uptake of the next analyte, and certain in parallel washing steps. So this is why nothing happens at this time. So we can just cut it out basically. So we just see the important part, which is the association measurement. And so, yeah, but you see the gap size is basically in real time. So you see the whole trace is basically what the real time trace. Yeah.
Okay, maybe I just quickly cut in with a small organizational question, because that also came up. You can watch this webinar afterwards. It will be on demand. We will record the webinar, which also contains the slides for people that maybe missed the first few minutes. But now back to the science part. People are also interested in the fluorophores that can be detected. Would you like to comment on that?
So,
you want to?
So we have the system is made, so the optical detection system is made for, has a green and a red channel. So they are like, it's made for all the standard fluorophores available on the market, basically. So yeah, standard green fluorophores, standard red fluorophores. So in the around 500 nanometers and 650 nanometers. Which can be ordered and yeah, which is like a standard tag for tagging the analyte.
Yeah, I think it offers quite a broad range.
Yeah, exactly.
Range of analytes, right?
Yeah.
Okay, now this is probably a question that many of you are interested in. Somebody asked how the kinetics correlate with soluble protein data, for example, with SPR data. Of course, we get this question quite a lot. So
maybe I can tell a little bit from our customer experience that we've had so far, because obviously this is a very important question. Like, is my SPR data actually enough? Do I need to measure on cells even? And I think there are two ways to answer this. I mean, there are samples which are basically giving exactly the same kinetic. I mean, straight out, we measure the same what was measured on SPR, but this is not always the case. So I would say it's absolutely relevant to measure the kinetic on cells to know if this is the same, because it doesn't have to be the same. It can be actually very different for all the reasons that we mentioned in the beginning of the influence of all the native environment on the cell, actually, especially just sterically, if the target of, for example, an antibody is not so accessible on the cell, it just slows down, for example, the association rate, even if it's then maybe having the same dissociation rate. So this can have a huge impact, and we are not always getting exactly the same values that are people reporting or giving us as a reference from SPR measurements or BLI or other measurements that they have. But I would say overall, what we can always reproduce is kind of the range. So what is the stronger binder? What is the weaker binder? Like this is normally comparable.
Mm-hmm. So now that we are already talking about comparisons between different technologies, of course, for many users, it's also important how fast you can be with different technologies. So this relates to a question about the duration, like how long does it typically take to measure a 96 or a 384 well plate?
So this strongly depends also on the interaction you would like to observe and characterize. So if you take, if you have a weak binder or something in between, which is often in around 30 minutes, let's say, then it for sure takes not that long as we, if we have look at like tight binding antibodies, for example, which needs like several hours until they are off the target. So this is strongly dependent on the interaction, but as a system is fully automated, it is very easy to handle, and also the whole procedure from harvesting the cells until the cell is trapped on the chip and the first concentration of analyte is going over the chip is basically done in 30 minutes. So after 30 minutes, you see whether it binds or not binds. And then there will be several concentrations of analyte and the dissociation, but the whole system is fully automated. The chip loader can take up to 5 chips with automatically chip change. So you can run it for basically hours and days fully automatically. But it's there's no like no time where we can say it takes so long. So it strongly depends on the interaction you want like to observe.
But I mean, since it's optimized, you could probably also just run it overnight.
Sure, overnight.
We always
even like 24 hours.
Stuff in between, yeah.
Exactly.
Okay, so now we get a bit more into the experimental details of specific targets. If the target receptor undergoes internalization, how does the signal change?
That is a very interesting question. I think now that we have the opportunity to actually look in the time course at single cells that are even living, it's not just the binding and unbinding of a molecule on top. Obviously, something happens on the surface and it depends on the target. This target might be internalized with the protein or with the antibody or whatever is bound to it. So it depends, what you observe then really depends on how you labeled your molecule, because we can actually detect the entire fluorescence no matter where it is basically in the channel. So it could also be within the cell. But if you have a pH sensitive dye on there as a label, it might react basically to the acidification when it enters the cell. And then your fluorescence signal will change. So if you want to, or if you expect internalization to happen, it depends on your question basically. If you would like to measure your binding kinetics without internalization, I would, for example, suggest to try to take that out in a measurement and measure, for example, on fixed cells, or to chemically stop the internalization, or measure the low temperature where you slow down all the physiological processes. So basically just isolate the individual processes that are taking place, look at the kinetics, and then see how the actual internalization is affecting that, so that you understand what is really happening here.
I mean, that's also a big advantage, right? I mean, just molecules cannot change their receptor expression. So since we are doing it on cells now, you have the whole picture, basically. It's really nice. So back to the technology. So this is probably a question for Sebastian again. Does the flow speed affect the K on and K off time constant? And yeah, maybe we start with the first half and we ask the second half of the question later.
Yeah, very good question. Since we can apply very high flow rates, we until 500 microliter per minute, we can basically, there are no, or we can completely, there are no mass transport limitation effects or rebinding effects. We can go very up with the flow rate at the one hand, but also for weak binders, the high flow rates are very important, that we really can wash out the bound analyte very quickly to resolve these fast interactions. So certainly we have flow rate ranges between 20 and 500 microliters per minute. So it depends also on the sample consumption. So it's a very flexible system, and you can adjust it between sample consumption or really powerful fluidics to get rid of mass transport and rebinding.
And this also, I think, leads to the second half of this question, which is how to set the applying duration for different concentrations. Actually, if I understand it correctly, that's basically like, how do I decide about my flow rates, right?
Yes, that's true. Yeah, what you want to reach is with the highest concentration, and you want to go in saturation, yeah.
Maybe from a practical point of working with customers, helping them setting up as is quite a lot. So I can just say this really comes down to a basic understanding of how to set up a biophysical assay here, no matter if that would be an SPR assay or a switch sense assay or here now RTIC. So this is always kind of similar. So what we are recommending is always to aim for a concentration range where the analyte is going like at least 10 times above the estimated KD. If we don't know the KD, we do a kind of screening assay with different concentrations, just find out in which range are we. And the flow rates, we are normally choosing rather low to start with, actually, because most of the time we are not seeing any technical artifacts. And I would say if you see an influence of the flow rate on your on and off rates, it can only be like a technical artifact that you see, because the flow rate itself cannot actually change your biophysical interaction parameters. They are just fixed constants, right? So if you measure something different at different flow rates, it means you were probably in the lower flow rate not fast enough and you had mass transport limitation or rebinding. So if we see these effects, then we would increase the flow rate. If we don't need to flow faster, because that would just consume more analyte. We don't want to consume more analyte. It's just, let's work at low flow rates and have only as little analyte in there as necessary, right? And then we have normally really nice, clean association dissociation rates.
Now that you're already talking about like analyte consumption, one thing that's also coming up is how many cells do I actually need per run? I guess this is also interesting for people working with like patient cells or primary cell cultures, which are very unique. So do we need a lot of cells?
I think this is, I don't know, it depends on the perspective. I would say no, not really. So each individual capture event takes 35 microliter from a cell suspension that we normally recommend to have it at a concentration of 1 million cells per ml. But it's 1 million cells per ml. We're not using 1 milliliter. We are using 35 microliter of that. So this is just a few thousand cells that you're actually using per measurement. So you don't really need a lot of cells to have a successful measurement.
Yes, that's good news. When we work with antibodies, there's a question about antibodies, whether it only works for them or if we also looked at FABs, for example.
Yes, we did. This is absolutely not limited to antibodies. Antibodies are very nice to work with. They're commercially available. So it's just a very good example for us to work with. And also a lot of customers are working with antibodies because they're using RTIC for the development of these antibodies. But FABs are just basically almost the same. I mean, they're part of an antibody with the advantage that we are having an easier setup because it is giving normally a one-to-one interaction. So we are always going for affinity and we understand the rates even better, like the biology behind it. So we can use also antibodies, we can use FABs, we can use proteins, we can use small molecules, we can use almost everything that you can label.
I think that's also like already the answer to the next question, whether it's also working for other formats like peptides. So as you already said, there are so many examples that you also already tried out with customers.
Yes, we did. Also peptides have been tried, yes.
Yeah, so it's only your fantasy, that's the limit. Just give us whatever you want to measure. Could more than one cell be trapped in the traps? And how diluted should the sample be is another question.
So we offer 3 different sizes of traps on the chips. So it is ideally, so we have a small trap, which is SML, so which is the largest one. So you should know about how big your cell is, so that just one cell is trapped in one cage, basically. Yeah, but it can be that there are also like 2 cells in one trap, but mostly it's, I think, just one cell in the trap, right? So
yeah, so this depends a bit on the choice of traps. We are not always knowing the size of the cells before exactly, then we would just maybe go with the standard, like the middle size or the large size, to be sure we trap something. And depending on the question, more cells in the traps don't even need to be a problem, because the more cells you have on there, probably you get the higher signal amplitude. That's also why we have a chip with only one cell trap on there, not really a single cell measurement, but we also have the chip format with 5 traps on the chip already, so that we can capture for sure several cells. And then the aim, of course, is not a single cell experiment, but for example, the average in the population. I just want the kinetics overall in this population of cells. I don't need to do a single cell experiment. I just want to have a good signal amplitude. I capture several cells, and that's great for me. And if the traps are perfect in size to have single cell in there, it's nice. But sometimes we have measurements where we have 2 or even 3 cells in there. That is not a problem. It's still washed well around cells then, and giving us a good signal. That's
and
important.
I think the important thing is also, you know what you have, right?
Exactly.
Since you control it, it's not like you never know how many cells you got. You can always like interpret your data. The next question is maybe also a bit related to the signal amplitude we were talking about. It's, do you need to have higher expression to be able to observe binding of smaller molecules?
It has nothing to do with the size of a molecule. So we are not measuring by size of molecules. There's a big difference, for example, to an SPR system where you would normally get a better signal by a larger molecule. We are completely independent of size. What we measure is fluorescence, and basically that is the signal readout. So if we have one fluorescent tag per molecule, whatever size the molecule is, that is all we need. And if we want to increase our signal, we can, for example, add several fluorophores per analyte. We can do that, but that's mostly not necessary. But of course, it depends now on how many target molecules I have in my cell, how many molecules I can bind actually to get a total signal. So obviously, a very low expression of the target will result in a smaller signal amplitude. And there is a lower limit at some point, which is probably somewhere, depending now of the labeling of the analyte and everything, somewhere in the range of 1,000 to 10,000 molecules per cell. But I would say if you have a very low expressing target, just let's try, and we can change all our parameters to be very sensitive there to get out a signal, even if you have a low expressing target.
And I mean, this was also nicely shown in the last example, right, where you had like the different expression levels and so on. So it's also nice to use this fact that you get a lower signal amplitude, because you can also get information out of that. Okay. So I hope I didn't miss any question. We've got another one about the fluorescence background noise. How can I diminish the fluorescence background from the actual signal?
So yeah, also very good question. The fluorescence, since we are working with fluorescently labeled analytes, there's a high background during the association phase. But this is not a problem, because we have 2 electrodes. One electrode is a sample electrode where the trap is on, so the cell gets trapped on there. So there the interaction takes place. And on the other electrode, there's no trap. This is a reference electrode. And by that, so we have a reference channel, and we can reference out the high fluorescent background. So yeah, this is yeah, super easy and automatically done by the software. Yeah, so no problem.
Okay. Now there's a question. Can you check cell-to-cell complex bound by antibody? I'm not sure if I understand that question correctly. I guess maybe it's like a cell-cell interaction that is initiated by antibody binding, or maybe you can a bit like elaborate a bit on this.
I'm also not sure how the question is meant.
Yes. So, I mean, in general, maybe we can, in general, talk about cell-to-cell binding?
I mean, that question comes up sometimes. That people would like to go a step further to look at a cell-to-cell interaction, because if it's, for example, a T-cell recruitment, that could be the natural effector in the end. We cannot do cell-to-cell. And there's, I think, a very important reason that we don't need to do cell-to-cell. There are technologies out there that can do that. But if you do a cell-to-cell measurement, you cannot calculate back to the affinity and avidity of one single interaction. So if you really want to understand the kinetics of one individual interaction and be able to modify that, for example, to increase a certain stability of binding, or to increase a certain on rate or decrease, you need to really first measure the individual interaction. Only when you understood the individual interaction, then I guess it makes sense to then go a step further to say, okay, now how does the effect of the density of all these molecules and everything come into play when it's really cell to cell? But then you would have to go to another device. This is not ours anymore.
Yeah, I think you can see it like as a nice combination. You start out with RTIC and measure the antibody binding to either cell, and then the cell-to-cell complex is going to be validated with a second downstream assay then afterwards, right? Mm-hmm. Okay, I hope this answers your question. Please tell me if not. The next question would be if there's also a way to correct fluorescence depending on the buffer we use. I guess so. Yeah, go on.
Understood it correctly. So we have normally we're using one running buffer. This can be supplemented with like complex media, for example, that living cells are feeling healthy in the buffer. So we have one buffer, and we have this labeled analyte, and we have one flow channel, and the buffer and the cells and the analyte is going over, flown through this flow channel and over two spots. So I'm not sure if I understood this question correctly, but basically, yeah, maybe can specify a bit more. What do you mean with this question?
I think like the core is that even if the buffer is autofluorescent, that doesn't matter for our measurement, right?
If it's about autofluorescence, like media, for example, it obviously creates a higher background, and I would maybe not recommend to go very high in a high fluorescent background. Even if we can calculate that out, at some point we are reaching like a limit of what we can then still see as a signal on top of that. But this has never been a problem so far. Meteor even exists without dyes in there. So this second electrode that we are automatically using in our measurements to simply subtract everything that is giving a fluorescent background from the buffer or from the analyte, whatever is in the flow channel, works really well. So this is nothing we need to think about.
So the specification of this question would be specifically DMSO. Have we worked with DMSO yet? So the person is working with a small molecule in DMSO, and that leads to many artifacts even after subtracting a reference.
So DMSO is not causing any fluorescent differences. So we can add DMSO in. I would maybe not use it in a very high concentration because that's maybe not very good for the cells. But if it's in a standard like 2% or whatever, like a low concentration in there, would not be any problem. But it has basically no effect on the fluorescence that we are recording. So there's nothing we need to consider with the DMSO. It's just in there. I know it can cause like signal jumps in SPR, for example, but since we are measuring completely different, we're just looking at the fluorophore, and we're looking at the same time at the measurement and the reference spot in the same channel. So it's not different channels. It's really sequentially at the same time measured. It just basically has no impact on the measurement.
Yeah. But also if you have very specific questions about your specific analyte or cells, you can also contact us afterwards, and we can also discuss this in more detail and even try it out if you want. So I will also tell you the email address afterwards, and just keep that in mind. You can always contact us for very, very specific questions. What are the limits of fluorophore in the buffer before you can't do the reference subtraction?
Yeah, so basically this is going into the same direction. What is the limit here now of fluorescence that we can generate in the channel? There is not a certain concentration, because what we can change as a parameter is the excitation power that we apply. So if, for example, I have a low affinity binding in a micromolar range and I need to use micromolar concentrations of my analyte, I can, first of all, use a very low LED power for excitation to not go into a too high total signal. But I can also, for example, decide to not label every molecule. Maybe I just use 50% or maybe even just 10% labeled as a tracking molecule, and the rest is just unlabeled, so it's not generating so much background. So there are always ways to deal with a high fluorescent background. This is normally not an issue.
So looking at the time, I think we have time for one more question, which is when kinetics are measured using the biocore, some people sometimes observe non-specific binding. Have we also observed something like this with our system? And how do we deal with? I guess every system observes non-specific binding, but the question is how do we deal with that, right?
Yeah, I mean, obviously there are different surfaces in the different materials, and depending on an analyte, it can adhere to that. For example, charged peptides like to stick to glass. We have a glass shell in there. It can, for example, stick to that then. First of all, we see that very clearly because we're so sensitive. So whenever something like that is building up, we can see that very nicely. So we're not blind to it. We're not just magically working with a lower concentration of analyte and don't know why, because we see that it is actually adhering somewhere and being like caught out of the solution. And what we do about it then are different approaches. So first of all, we can passivate surfaces. The gold surface is passivated, but we can, for example, use standard buffer additives like a BSA to just passivate also the glass if it's necessary. We're not standard doing that, but if we see that this is required, we can do that. For something like if it's charge related, we can increase the salt concentration a little bit in the buffer to prevent that. And this is normally then already solving the problem. So I would always look at the individual sample, trying to figure out why is this sticking basically. And then based on that, trying to improve it. Yeah.
And does it happen very often? I think
it's completely analyte dependent. In many projects, it's not happening at all. You inject, it's nicely injected and immediately washed out, no sticking at all. But it can happen sometimes, yes.
Yeah. But it's not something you have to like always be aware of. Okay, so yeah, I think it's time to wrap up this session already. Thank you all very much for participating and for the lively discussion. And thank you to Nina and Sebastian for your insights. And if you have any further questions or comments, please just send them to us to info@dynamic-biosensors.com. And why not just try it out, the heliXcyto? You can book a technology demonstration with us. You can just contact us or also go to our website, dynamic-biosensors.com. And make sure you follow us on social media like Twitter and LinkedIn to always stay up to date with all the current developments. So thank you all very much and have a great rest of your day and goodbye.
Bye.
Bye.